Hello and welcome to the B31 review video on gas exchange. Now this is a true IB unit right here. When we say gas exchange, I'm sure the first thing most of you are thinking is, lungs. But no, we're IB here. Okay? This is leaves and lungs. A little bit of both for you. So gas exchange is vital for all organisms, all, including bacteria. But it's a lot easier for single-celled organisms, right? They can just exchange gas through the surface, and so, but their surface. Now, they're affected, and so are multicellular organisms, but they're affected by surface area to volume ratio. This affects everything. You need a high surface area because your ratio of surface area to volume, they're both going to increase, but surface area is squared, volume is cubed, and so the volume will increase faster. So the rate of removal and entrance of gas will not keep up if the cell gets too large. Keep up. And so there are multiple things you can do to affect it, right? So if you just go like this, you can see how, not a perfect example, but like the distance from the center is increasing and the amount of membrane is not scaling as like the total volume is. So what do we do? Well, gas exchanges primarily all are the same in both animals and plants. They have to have these capabilities. They have to be permeable. So they have high permeability, right? So they have high permeability to allow things to move in and out, in and out. They have a thin tissue layer, right? So this minimizes the distance that it has to travel. So lowers travel distance. They also typically are a moist surface. This moist surface helps increase diffusion of gases. Helps increase diffusion of gases. And then of course, they have to have a large surface area. How can we get a large surface area? Well, the easiest way, of course, is like folding, right? So adding folds to increase the surface area. Now, let's talk about some animal stuff. How do we maintain concentration gradients? So for a gas to move from one spot to another, it must be it's diffusing, right? So it's got to go from high to low. Well, how do you make it from high to low? Well, you move the other stuff this way. And so this is our blood or our circulatory system. And so what you can do is pump the blood, and that will pull the oxygen away, right? And it will also bring the CO2 in, and so it serves a dual purpose, to remove oxygen from near the lungs and bring CO2 to the lungs, where it can be expelled. The other thing you can do is, if you're a fishy, you have your gills and you swim, water will go along it, right? So you have your, this would be a lot better with another color, sorry. You have your blood vessels in here, right? So this is blood. This is water. And so the movement of the water is going to bring more O2, and then the blood will carry it away. So the movement of the water through the gills when they're swimming helps maintain a concentration gradient. So this is all about this concentration gradient. For humans, not only do we have the blood, but we also have our lungs. And so you take air in and air out. And so this keeps also helps keep a high concentration of oxygen. Now, how do lungs work? So there's two things we need to know, which is one, what are their adaptations, and two, how do we actually move them? That's the next standard. And so for this, we only care about mammal lungs. And so mammal lungs are like ours, and it looks like they got little grapes. Grape, 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 grape. And so those grapes are connected to a whole bunch of other grapes, grape, 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 like that. And so these things here are called bronchioles. And so if we were to zoom out, you have your trachea, your bronchi, and then each of those breaks. Trachea, bronchi, and then here we have the bronchioles, and at the end we have the alveoli. You can see the bronchioles help give you a whole bunch of alveoli, and then the alveoli, right, have really high surface area, right? So they have all these folds which give them a high surface area, and they also are covered in capillaries. So capillaries are our thin blood vessels, and so you're going to cover the alveoli and blood vessels. Cover, because I can't actually draw it, sorry. I'm not that talented. I know, I'm sorry. And so then what we have happen, that is like how the lungs actually look. Then the other thing we mentioned is that we can ventilate them. So when we think of breathing, that's the IB word, is called ventilation. And so there are a couple of things that happen. Step one, your diaphragm contracts and flattens to pull the lungs down. Then your external intercostal muscles pull the ribs and lungs outward. This increases the volume, which, pivnert, decreases pressure. And that decrease in pressure creates a vacuum effect to pull air in. Then, so that is inhalation. Exhalation is the opposite. Your ab muscles contract. All of your muscles work in pairs and pulls lungs up. So that one must work opposite the diaphragm. So if this is contracted, the diaphragm must be relaxed. Then the internal intercostal muscles pull the lungs in, which causes the volume to decrease, which causes the pressure to go up, pivnert again, which pushes air out. So all of this is to help maintain that concentration gradient that we saw on the last standard. So that is how ventilation works. Now, how do we know the different quantities in your lungs? So you have 4 things. You have your tidal volume, your vital capacity, your inspiratory reserves, and your expiratory reserve. You can measure all of these with a spirometer. Spirometer. There we go. Okay, all of these can be measured with a spirometer. What is a spirometer, you ask? That's a great question. A spirometer is a device that you like blow into and you go and it changes it for you. Okay. Now, these different things, the tidal volume is your normal breath. It's how much air you expend normally. Your vital capacity is like the maximum you can breathe in and out. So the maximum of your breath. The inspiratory is the amount you can take in more than tidal, and the expiratory is the amount less than tidal. So if you go, that would be the expiratory, and if you take in a breath, and then you take in a little more, that would be like the inspiratory. And so your normal breathing looks like this, but you can also go above that, and you can also go below that. And so that's tidal right there. The total would be your vital capacity. This would be the N, this would be the X, and the whole thing would be the VC. So this is TV, so tidal volume, that's the inspiratory capacity, reserve, expiratory reserve, and then the vital capacity is the whole thing. Okay. That was standard, I didn't say, sorry. Standard 6. Standard 7. Oh no, oh, sorry. I just have to draw a leaf, and I got sad. Can we draw a leaf? Yeah. So leaves look like this. Ta-da, I did it. Wait, this looks like the cell membrane. Yes, it does. So you have here the epidermis, the epidermis layer. So I'm doing standard 8, because you should be able to draw this. And it's also 7 at the same time. So this top, there's also on top of the epidermis is a waxy cuticle. The waxy cuticle and the epidermis both prevent evaporation, because plants don't like losing water, so they prevent evaporation. Then they have some like palisade mesophyll cells, and these are going to have lots of these organelles in them called chloroplasts. And so those chloroplasts are where photosynthesis is going to happen. Then you have your vascular tissue running throughout the leaf, right? So that would be vascular tissue as well. Then on the bottom, you have guard cells. And then if they open and they go turgid, you have your stoma, or an opening. We also have what are called spongy mesophyll cells, and they look more like this. And there's some kind of like gaps between them. And so these cells are the spongy mesophyll, and they allow for gas exchange. There's also just air spaces, right? So this is an air space. And so that also allows for gas exchange. And so that is standards 7 and 8. That is the different adaptations. The veins, of course, that look a little fancier than this. They look more like this. And so they have the different kinds in the vascular bundle. And they're going to, of course, move. In this case, they're moving water. They don't have blood, right? They have a water-dense thing, and they have the xylem and the phloem, which we'll talk about eventually. Okay, that was 7 and 8. Means we need to do number 9. So transpiration. So when the stoma is open, water will evaporate out of the stoma. And that is called transpiration. So transpiration is the evaporation of water from a leaf, or from a stoma. Well, why is that happening? So it's like you got all these water molecules, right? And as they leave, they're going to pull more up with them. That is a terrible water molecule. But anyway, they're evaporating. Now, there are things that affect evaporation, right? So things that affect evaporation are temperature. Higher the temperature, the more transpiration. The humidity is a weird one, so I really shouldn't do it next. So I'm not going to. Light. The more light there is, the more transpiration. The more wind there is, the more transpiration that will happen. Because these are all like increasing temperature. This is going to cause them to open the stoma more, therefore there's more light, more evaporation. Wind is going to pull those molecules away more. And it also, wind typically lowers the relative humidity, and humidity works in the opposite direction. The lower the humidity, the more transpiration. And this one has to do with concentration gradients. So the higher the difference between the leaf and the outside, the more evaporation will happen. And so if it's really humid, not a lot of evaporation will happen. So that's why desert plants need to have adaptations to prevent water loss more than your like jungle plants because of the differences in humidity. And so, for example, they can do things like open and close the guard cells in the desert to stop water from being lost. Okay, great. Stomal density. You should be able to look at a leaf and look for these things and count them. And then you take the number over the area to get the density. Yes! And that is the end of the SL. Alright, HL only. There are only 3 standards, which is nice, but they're kind of annoying. So let's talk about them. Alright, so the first one here is all about hemoglobin. Hemoglobin is a protein. It's a quaternary protein with 4 chains, and each chain has a prosthetic group with the heme or the iron attached to it. Iron has a really high affinity for oxygen, and so oxygen will bind to that iron. That's the first adaptation it has. It also, when it binds the first one, it induces what's called a cooperative effect, and it allows it to bind even more. And so it allows it to bind lots and lots of oxygen through the cooperative effect. That is 2 of the adaptations. It also, one of the adaptations is like different species have different affinities, and so their hemoglobin can look a little different too. Fetal hemoglobin has a higher affinity than our adult hemoglobin. Why is that important? Well, let's think about it, right? If this is like fetal blood, and this is the mom, and they're passing like this, these cells need to be able to steal the oxygen away from the other one. And so they have to have a higher affinity for oxygen so that oxygen will actually transfer from the mom to the fetus. Okay, cooperative binding. Oh, allosteric binding of carbon dioxide. So for the allosteric binding of carbon dioxide, this is going to change the affinity as well. And so they have an allosteric site. Remember, allosteric site are for inhibitors, and it actually lowers the affinity, and this is in both adults and the other one, and fetuses, lowers the affinity for oxygen. Well, what that's going to do, so CO2 will attach to the allosteric site and lower the affinity for oxygen, which is going to cause oxygen to be released to the tissue, which the tissue needs it, right? Because it's low on CO2, or sorry, it's high on CO2, therefore it must be low on oxygen. Our body is really bad at sensing lack of oxygen. It's really good at sensing high amounts of CO2. And so what happens is, this is actually standard 12 as well, it's called the Bohr effect, or Bohr shift. And so what the Bohr shift or Bohr effect does is CO2 will bind to hemoglobin, and it will cause it to release the oxygen, thus helping respiring tissues. And by respiring, we mean ones that are doing cell respiration. And so this is then going to cause oxygen to be dropped off where it needs to be, instead of just randomly dropped off at different spots. And so when you're working out, right, if you're working out your arms, that will cause more CO2 to be created in your arms by your muscles, which then will cause that to bind to hemoglobin in the allosteric site, which will change the conformation, which will release the oxygen. Therefore, your muscles get oxygen, so they breathe and you don't die. Yay! All right. Then the next standard we need to know is our oxygen dissociation curves. And so our oxygen dissociation curves, they look like this. Where'd my pen go? Oh, here it is. It's an S-shape curve that looks like this. Okay, so what does this graph actually mean? Here we have the pressure of oxygen, or the partial pressure of oxygen, excuse me. Over here we have the hemoglobin saturation. So how much of the hemoglobin? At the top we have 100, at the bottom we have 0. And you can see that it spikes up relatively fast. Honestly, this probably isn't quite spiky enough. And so what happens is it goes up pretty fast and then it levels off because of the cooperative binding effect. Now, you can shift it so where you need more oxygen to get the same effect. This would be the Bohr shift. You've shifted it this way. So when CO2 binds, the affinity goes down. So the farther this way it is the higher the affinity. So affinity goes up as it moves this way. So a curve like this would have really high affinity. So like this would be like fetal versus adult versus the Bohr shift. And so it's the ability for it to attract oxygen and become saturated with oxygen. It does level off near the end, right, as the oxygen gets packed in there too.